Welcome to this explainer. Okay, today we are diving into something truly fascinating, but yeah, honestly, kind of terrifying. We're going to take a really deep dive into a microscopic subject that can turn a perfectly innocent summer swim into an absolute medical mystery. We are talking about Naegleria fowleri. You probably know it by its more infamous name, the brain-eating amoeba. Now, it is a rare pathogen for sure, but the clinical progression is incredibly rapid, and the underlying biological mechanics of how it actually infects the human body, well, they are just astonishing.
Just four. Out of 151 documented cases of this infection in the United States between 1962 and 2020, only 4 people have survived. Think about that for a second. That is a staggering mortality rate of over 97%. And when this pathogen strikes, the median time from symptom onset to death is just 5 days. Literally 5 days. It is a true race against the clock, which makes understanding this organism absolutely crucial.
And look, this isn't just some static issue from the past, it's actively evolving. Microbiologist Charles Gerba put it pretty clearly. He said, as the temperature of surface waters increases further north, we expect more cases in the future. We're observing a real environmental shift here. Naturally warming waters and changing global ecosystems are objectively expanding this pathogen's geographic habitat. We are starting to see it pop up in regions where it simply wasn't found before.
Okay, let's dive into this. Here is our roadmap for today's explainer. 1. The expanding microscopic threat. 2. Invading the central nervous system. 3. Weaponizing the immune system. 4. The race to diagnose. And finally, 5. Treatments and prevention strategies. Let's get right to it.
Section 1. The expanding microscopic threat. To really wrap our heads around this amoeba, we've got to first understand the specific environments where it thrives. Before we can look at how it interacts with the human body, we need to know exactly where it waits.
So the most crucial thing to know is that Naegleria fowleri is incredibly particular about its environment. It literally loves the heat. It has optimal growth at 40 degrees Celsius, that's about 104 degrees Fahrenheit, and it can survive in temperatures all the way up to 46 degrees Celsius. Because of this, you'll primarily find it in warm, stagnant freshwater lakes, hot springs, and slow-moving rivers. Now, it absolutely hates salt, so it loses activity and just dies in marine environments like the ocean. Instead, it spends its time hanging out in the muddy sediment at the bottom of fresh water, feeding on cyanobacteria. Interestingly, extreme weather really alters its spread. Droughts can actually concentrate the pathogen in shrinking water bodies, while on the flip side, extreme flooding can displace it and bring it much closer to residential areas.
And this brings us to a really significant geographic shift. Historically, these cases were almost entirely contained within the warmer southern United States. But as we mentioned earlier, those boundaries are changing. We're now seeing infections steadily expanding northward into new frontiers, places like Nebraska, Iowa, and even as far north as Minnesota. As the surface temperatures in these states warm up during the peak of summer, recreational waters that were previously safe are becoming perfectly viable habitats for the amoeba.
Section 2. Invading the central nervous system. Now let's shift gears and zoom way in to the microscopic scale. Let's put on our medical detective hats for a second. How exactly does an organism living quietly in a lake end up destroying a human brain?
Let's move to and see how this builds. It is a shockingly efficient 4-step sequential journey. It all starts when contaminated water is forcefully pushed up a swimmer's nose. First, the amoeba adheres tightly to the nasomucosa using this highly specialized structure called a feeding cup. Second, it crawls directly up the olfactory nerve. Third, it hits a bone called the cribriform plate, but get this, the amoeba actually secretes specific enzymes called matrix metalloproteinases to literally degrade the tissue and chew its way straight through the bone. Finally, it infiltrates the subarachnoid space, which is the fluid-filled area surrounding your brain, and enters the brain parenchyma itself, the actual functional tissue of the brain.
You know what's truly terrifying about this? The amoeba is essentially acting as a microscopic hitchhiker. The human body has incredibly robust defenses to protect the brain, right? Most notably, the blood-brain barrier. But Naegleria fowleri doesn't even bother trying to cross that heavily guarded wall. Nope. By hitching a ride directly up the olfactory nerve, the very nerve that controls your sense of smell, it exploits a direct anatomical backdoor straight into the central nervous system.
Section 3. Weaponizing the immune system. Once inside the brain, the infection takes hold. But here is the most counterintuitive and, frankly, tragic part of this entire process. Yes, the organism itself does damage, but your body's own defense mechanisms actually cause the most catastrophic destruction.
And this brilliantly illustrates the amoeba's highly specialized toolkit for destruction. Let's look at its virulence factors. First, the NBFA1 protein helps the amoeba adhere to and physically consume host cells. Then you've got these pore-forming proteins, which do exactly what they sound like. They punch holes in the host cell membranes, causing them to completely explode and die. Finally, there's cathepsin B, an enzyme that degrades the structural matrix around the cells, severely altering the stability of the blood-brain barrier from the inside, which allows even more amoeba to just pour right in.
So, as the amoeba attacks, the brain obviously screams for help. And this leads us to necroptosis. This is a severe form of cell death triggered by an overwhelming inflammatory response. The body basically panics and releases massive amounts of reactive oxygen species to try and kill the pathogen. But in doing so, it triggers an overzealous defense force that accidentally destroys its own city. The resulting brain swelling and tissue necrosis are incredibly rapid and severe.
This overreaction creates a full-blown cytokine storm. Let's quickly look at the cells that get hijacked into causing this uncoordinated damage. Microglia, which are your brain's vanguard immune cells, end up releasing highly neurotoxic substances. Astrocytes go into total panic mode and recruit massive amounts of other immune cells into the confined space of the skull, causing lethal pressure. And then neutrophils deploy what are called nets, literal biological traps meant to catch the amoeba. The problem? They almost entirely fail to eliminate the pathogen, but they cause severe localized inflammation that destroys the surrounding brain tissue anyway. It's just devastating.
Section 4. The race to diagnose. Because the brain is swelling so incredibly fast, and that median survival window is just 5 days, the absolute most critical phase of clinical intervention is the race to diagnose. Early clinical suspicion is vital, but the really tricky part is that the early symptoms perfectly mimic common bacterial meningitis.
So here is the intense diagnostic dilemma doctors face. You basically have low-tech versus high-tech. Taking a sample of cerebrospinal fluid for direct microscopic examination is fast, it's direct, and it's cheap. The problem? It's highly prone to false negatives because the amoeba can easily hide in the sample. On the other hand, we have metagenomic next-generation sequencing, or MNGS. Now, this has extremely high accuracy and can sequence the genetic material of rare pathogens directly. But it's highly expensive, it takes precious time that the patient just might not have, and it's very susceptible to sample contamination.
Section 5, Treatments and Prevention Strategies. So if doctors do manage to diagnose it in time, how do we actually fight back? Let's pivot and look at how modern medicine is attacking this microscopic threat, and honestly, more importantly, how you can easily protect yourself.
Currently, the medical field faces some pretty severe treatment bottlenecks. We are mostly relying on what you'd call old drugs with new uses, primarily heavy antifungals and antiparasitics like amphotericin B and miltefosine. But remember that blood-brain barrier the amoeba smartly bypassed earlier? Well, it successfully blocks our medicines from getting into the brain at high enough concentrations to work. To compensate for this, doctors have to administer massive, highly toxic doses. They are forced to walk this razor-thin line, literally having to choose between killing the amoeba and poisoning the patient's kidneys and liver.
But there is incredible hope on the horizon here through next-generation nanomedicine delivery. Scientists are actually engineering nanocarriers designed to intelligently sneak drugs past the blood-brain barrier. They're using methods like receptor-mediated transcytosis, where nanoparticles essentially have specific keys to unlock barrier receptors. Or, my personal favorite, the brilliant Trojan horse approach. This involves literally hiding microscopic drug particles inside the patient's own immune cells as they naturally migrate into the infected brain. This could deliver a lethal payload directly to the amoeba while totally sparing the rest of the body's organs. How cool is that?
But hey, until those sci-fi nanomedicines are widely available in every hospital, prevention is absolutely your best defense. And if we tie it all back to the science we just covered, these rules make total sense. Number 1, avoid warm, stagnant freshwater in the late summer. Number 2, wear a secure nose clip. Remember, the amoeba must travel up the olfactory nerve, so blocking the nasal cavity completely shuts down its only entry point. 3, don't stir up the muddy sediment where it feeds. And 4, if you use a neti pot or do nasal irrigation, never ever use tap water. Use only boiled or distilled water. It's a simple step that literally saves lives.
We've explored the perfect storm of environmental conditions, the unbelievable biological hijack of the human nervous system, and the cutting-edge science racing to catch up. The story of Naegleria fowleri is a powerful, sobering reminder of our deep and sometimes vulnerable connection to the microscopic world around us. So I want to leave you with this thought. As our global ecosystems rapidly change and waters continue to warm, how will humanity continue to adapt to these invisible, migrating threats? Thank you so much for joining me in this explainer. Stay curious, stay safe out in the water, and I will catch you in the next one.